Hey everyone, we're going to talk about the process of mitosis, which is kind of the middle part of the cell division cycle. So this is happening after interphase, which means that the cell has already produced an extra set of DNA, it's produced extra organelles, and otherwise prepared for the process of splitting into two new identical cells. Now it's important to understand how mitosis fits into this, so to start, I'll give you a definition. Mitosis is specifically the division of the cell's nucleus. So it's just the splitting of the nucleus, and it's important to keep that in mind as we understand sort of the goals of the process of mitosis. Now, mitosis fits right here in the middle of the cell division cycle. So if we think of this as the entire cell division process, we began with one cell, which we call the mother cell, and at the end we're going to have two identical cells to that original cell, and we call those daughter cells. Now, not pictured in this is the actual nucleus itself, but the process here in the middle is how we go from having one nucleus to having two nuclei, and at that point then we're ready to end the process with cytokinesis, which is the actual splitting of the cell into two new cells. Mitosis is split into four primary phases that we're going to go through. The first one is called prophase. Now the process of prophase involves first the DNA in the nucleus actually condenses down into chromosomes. So sometimes when we represent cells, we draw these little X's showing the chromosomes that exist inside of the nucleus, because we talk a lot about chromosomes, how we get 23 chromosomes from your mom and 23 from your dad. For people, you have 46 total chromosomes. They're important when we talk about genetics, and important certainly to the process of mitosis and to all of biology. But interestingly, they only actually exist in that chromosome, that little X condensed form, in the middle of cell division. Otherwise, we don't have chromosomes. We have more of this mess of DNA, this tangle of just strands of DNA existing in a form called chromatin, which is still kind of packaged and managed, but it's much more sprawling and much harder to keep track of. And so during this process of mitosis, where we have doubled the amount of DNA, because we're about to make two new cells, we need one set for one cell, one set for the other. So we have twice as much DNA. If it was in this tangled mess, it would be so difficult for the cell to be able to manage and figure out how to get half of the total amount, not just half the total amount, but the exact right one complete set over here and one complete set over there. That'd be very difficult to manage. And so what happens is the genetic material condenses into these chromosomes wrapped up with these proteins, and it makes it much easier for the cell to then grab these individual chromosomes and start pulling them around the cell and moving them appropriately to where they need to go. So the chromosomes condense into these little X shapes. The nucleus then actually breaks apart, and that might seem surprising because the whole point is that we want to get a new nucleus. But in order to start moving these chromosomes around and to get them to opposite sides so that we can make two nuclei, we're going to have to break the one we've got. So the nucleus itself actually breaks apart, and here you can see that happening. And then the last thing is that we start getting these spindle fibers, and these are produced by the centrioles, which are an organelle. The centrioles move to opposite sides of the cell, and they get ready to then produce these spindle fibers, which are just protein strands that grab onto the chromosomes and can start moving them around the cell. So that's the process of prophase. Moving on, then we have metaphase, and I like to think of metaphase because metaphase lines up the chromosomes in the middle. So think middle for metaphase. The chromosomes get lined up by those spindle fibers right in the middle of the cell. And these X-shaped chromosomes are actually replicated pairs. So they exist in these little X shapes here, and I'm putting my knuckles together to make that little X so that I can show how they're going to get pulled apart. Now, what this actually represents is two identical copies of the same chromosome put together. And that's how the chromosomes align after the process of replication, and as they condense, they come together. These are actually things called sister chromatids that come together, and it makes one replicated chromosome. Now, the point of this is that for a human cell, we would have 46 of these X's all the way across the cell. And after metaphase, we're going to see those get pulled apart, which should make sense, right? Because we want, if a normal human cell has 46 chromosomes, then in the middle of this process we're going to have twice as many as that, so 92 total chromosomes, and so it's going to be 46 of these X's that get split in half. And that makes sure then that the bottom half of the cell and the top half of the cell are going to end up with the right numbers. So since I'm already talking about it, let's move on to that phase, which is called anaphase. And I think of anaphase as moving apart. So anaphase, the chromosomes are moving apart. And here that's what you can see, that the sister chromatids, these individual copies of each chromosome, are getting pulled to the top of the cell and the bottom of the cell. And that, as you can probably guess, that elongation and pulling genetic material to the top and to the bottom is just getting ready for us to split this cell right down the middle, so that way we have two cells that each have a full set of DNA, one on the top, one on the bottom, and now we can actually create two cells. So during anaphase, the spindle fibers are pulling the chromosomes apart. That takes us then to our final phase, which is telophase, and this is the point at which we end up with two nuclei. So during telophase we get two nuclei, and therefore we've completed the process of mitosis. Since mitosis is the division of the nucleus, we started with one nucleus. During prophase we broke that down, we moved the chromosomes, we split them apart, and now we have two new nuclei at the end of telophase. Another thing you'll notice is that the chromosomes begin to unwind. We don't need them anymore. Now we can spread them back out. They're getting ready to just be the normal DNA that they were, so that they can undergo the normal DNA processes that they do inside of the cell. And as you can see on the top and bottom here, we kind of have two fairly normal looking cells now. In the middle though, because they are still attached, in the middle we have some proteins that are beginning to sort of squeeze this apart so we can pinch it off into two new cells. And that's going to be the process that happens after mitosis, which is called cytokinesis, and that's actually sort of the karate chopping of the cell right in the middle, so we split into two identical cells. So the process of mitosis, just as a quick recap, we started with prophase, and that was the process by which the nuclei breaks down, the chromosomes are condensed, we start getting those spindle fibers, we line them all up in the middle during metaphase, we start to pull them apart during anaphase, and then we reform two nuclei and we finish mitosis during telophase. And at that point then, we're ready for the final step of cell division that occurs after mitosis, and that's that karate chop of cytokinesis. So that is the process of mitosis, and if you have any questions, let me know.